Hello, everyone. Today is a really special seminar for many reasons. It's actually the last seminar before the August summer break. And it's Regina's birthday, which I think at IGC everyone should know. And as I wrote in my email, today we have a very special guest, Thomas Bosch, who's a long-time collaborator with us at IGC. He's one of the coordinators with Luís Teixeira in the SymbNET network. So today the seminar is also open to the SymbNET network. And Luís and I, together with Thomas Bosch, Margaret McFall-Nye, and Martin Blazer, we are hosting alumni meeting for the summer course on Host-Microbe Symbiosis that we usually have at the Gulbenkian every other year. And this year we decided to do an alumni meeting via Zoom for the obvious reasons, right? But so I'll just move on to introduce you, Thomas. It's really nice to have you again at IGC, a bit far away this time. And Thomas is a professor in zoology at Kiel University. He did his original studies in Munich, also part of it in UK. Then also a 2-year postdoc in California. And then he moved into several other universities in Germany, including University of Vienna. And he's been in Kiel since 2000. He's the head of the interdisciplinary Kiel Life Science Center since 2013. And in 2016, he started a collaborative research center called The Origin and Function of Meta-Organisms. He's very interested in how the microbiota and the host co-evolve. And his research is focused on the understanding of the microbiota in animal development. And for that, he has established a really beautiful model system that you can see already in your screen with the Hydra that he's going to tell us all about it, where he can really study the causal effect, direct effect of the microbiota into the development and other aspects, including behavior of this animal. So with that, I'll just pass the word to Thomas and go ahead.
Thank you so much, Karina, for these flowers and the introduction. It's a great honor to be at the Gulbenkian. I would love to be there in person and not sitting in my country house, but time will change that next year. Hopefully we will be back in Lisbon and at the Gulbenkian. I'm talking today about observations we made in the last 4 or 5 years, and I never have presented it in public, and it's a great honor and also fun, and I'm a bit anxious how I get the complicated story over to you. And the complicated story started at a coffee break at a Gordon conference, and a lady addressed me, which I never have seen before. I gave a lecture at that Gordon conference in 2017, and Jana Nafrot approached me in the coffee break and told me that she may have an approach to solve one of the problems which I presented and open questions which I presented during my talk. And a few weeks after we met at a coffee break, Jana was in my lab in Kiel, and we did experiments. And then we joined up with a professor of mathematics, Professor Eva Kanzo at UCSC, and we developed a nice story. And the story is that animals have spontaneous contractions. Independent where you look for, all kinds of different animals, cnidarians and Hydra, worms. This is a zebrafish larvae here. Here is a mouse gut. They have spontaneous contractions. They're found everywhere, and they're also found in humans. This is a movie my friend Stefan Schreiber gave me. So what we mean with spontaneous contractions is it moves back and forth and it goes spontaneously. And it obviously has nothing to do with food transport, because the mouse gut you just saw sits in a ringer solution and is empty of any food, and it just continuously contracts. And the question we were addressing is, what drives these contractions? And has that anything to do with the microbiota? And of course, we had, and there was particularly to the first question, what drives these contractions? There were already quite good evidences that contractions are driven by intrinsic pacemakers. The question is, what type of cells is that, particularly in evolutionary old animals? But then we ask also, are these contractions in some manner dependent on microbial colonizers? So that's a multidisciplinary collaboration between fluid physicist Jana Nafrod, a mathematician, Eva Kanzo, and my colleagues Alexander Klimovic, senior postdoc in my lab, and a graduate student, Christoph Gietz. Of course, I never learned anything else in my life but Hydra, so we do it in Hydra. And it's a beautiful system because it's actually transparent. Here, with the help of some visualization technology, in green, you see the nerve cells. In red, you see the bacteria. And what we have shown first is some time ago that nerve cells are in direct interaction with the microbiome. The microbiome lives outside the ectodermal epithelial cell in the glycocalyx. It's quite diverse, but still not as diverse as in a mouse gut. Many different kind of bacteria. We can culture them. Nowadays we can visualize them. We can genetically manipulate them. So it's a pretty nice system. And this microbiome interacts with nerve cells, because nerve cells stick through the epithelium. They sit beside epithelial cells, and they go with their protrusions directly in the mucus layer where the microbes are. That was a big surprise some years ago. I know this animal for a long time, and I always asked myself, why do neurons and sensory neurons sit along the body column? There is no food. What happens there? At that time, we could not see the mucus. Now we can see this glycocalyx, and now it's very clear that these nerve cells stick through in the glycocalyx where the microbes are. And in this nice paper with a former postdoc, René Augustin, we have shown that the nerve cells secrete antimicrobial peptides, which are also neuropeptides, and they shape the microbiome along the body column. The neurons, this is the nucleus of the neuron. These are then single peptides, single vesicles with peptides, which you can see are going directly outside the animal and in the glycocalyx. And if you genetically modify animals by knocking down this antimicrobial peptide, you drastically change the microbiome. And from that, we came to that conclusion that this neuronal derived antimicrobial peptide has something to do with controlling the microbial colonizers. For us here, it's only important to keep in mind neurons interact directly with the microbes. Now comes the question, do the bacteria contribute to these spontaneous contractions? And this is a former PhD study. It's also published by a wonderful PhD student, Andrea Mugello. She's now postdoc at the Max Planck Institute of Evolutionary Biology. And Andrea first asked these questions. So can we somehow quantify and measure these contractions? And second, has it anything to do with the microbiome? And so Andrea developed a recording device for quantifying, and we had camera programs which then did all that quantitatively. And so we measured the contractions in time, the amplitude and the regularity of the contractions, etc., etc. So we could all get that very nicely. And we found, and that's published some years ago, we found that if you remove the bacteria outside the animals and completely make germ-free animals, you drastically reduce the number of contractions. And you can rescue that nearly completely if you give back the normal set of microbes. It does not work with monocolonized animals, and it does not work with a semi-main colonizer mixture, not so nice. You need the whole mixture, and then you get the contractions back. That was pretty surprising, fascinating. The mechanisms are still not completely understood. It's clear that's not only the number of contractions which go down, but also the regularity of the contractions. So normal contractions go pretty regular, but then in the absence, it comes very irregular. So we came to the model that outside Hydra sits a diverse microbiome. It needs to be diverse, as I said. Single colonizers don't do the job. They obviously interact with each other, and they seem to produce something, and that something interacts with a neuroreceptor, and this neuroreceptor activates the epithelial muscle cells, and that leads to the contraction. We still don't know this something, and people want to ask me, we are hunting for it. It's pretty complicated, and it's even more complicated because it comes from more than one bacterium. So I'm in good hope that we get it at some point. Thinking beyond Hydra, that actually is not something which my colleagues in our center, John Baines and others, would fall from the chair, because when they watch their germ-free mice, they know they have a reduced gut motility. And so the same thing, if you measure spontaneous contractions in the gut, germ-free animals have a much reduced spontaneous contractile activity. And there are nice papers in different, that's well documented. What about human? In human, increasing one of these modern lifestyle diseases is constipation. And actually, also Alzheimer patients, 10 years, because the patients get Alzheimer's syndromes, they have constipation. And constipation means that you have problems, irregularities in the spontaneous contractions. And that's not a very rare disease, 10 to 25%. Causality and mechanisms at that point were not known. However, if you transfer microbes from patients with constipation into mice, in germ-free mice, then you will induce gastrointestinal dysmotility. So something from these bacteria, which comes from human patients, is there, probably metabolites, which interfere with the mouse enteric nervous system and causes the mouse gut contractility to not function normally. So that what Andrea observed in Hydra seems to be a pretty common phenomenon, and we were looking how to study that. The first question was, can we identify the neuroreceptor? And this is also a kind of a very nice story for the junior generation, because it again goes completely interdisciplinary, thinking that concepts which are important are conserved in evolution. Nothing in biology makes sense except in the light of evolution. We teamed up with an expert in inflammatory bowel disease, Mauro D'Amato. Mauro has sequenced 11,000 Swedish people with inflammatory bowel disease, twins and patients and asymptomatic patients. So he has the most complete data set, genetic data set on inflammatory bowel disease. And since we know that that was associated with a reduced contractile activity, we asked, can we find some of the signatures of inflammatory bowel disease, genetic signatures, in Hydra? And if so, can we identify the cells? Can we study the function? Can we study the function, the genes, etc., etc.? So this work was done by Alexander Klimovich and by Christoph Gietz, and it's published last year. So we used this enormous genetic database from humans, and we looked at all these tables, and then we stumbled and picked 4 ion channels, among them ANU1, TRPM8, and SCN5. And we thought that these are good candidates, which may be very important, not only in inflammatory bowel disease, but maybe conserved. So we looked at that, and I make a very long story very short because all of that is published. And so we found not only the genes, we identified the cells, distinct cell populations, which express their genes, but we also did function. And when we blocked these ion channels, then we reduced contractibility. And so there are standard chemical interference drugs, which are very specific for these channels. So these are the pacemakers, which are driving the contractions, and they are very conserved. They are conserved from Hydra to mouse and man, and they consist of a limited set of ion channels, ANO1, TRPM, SCN, and some others. They also have some other receptors, which we have described in that paper, and all the ancestral pacemaker cell, which also you will find in the mouse intestine, is prone to interact with the microbiome and to recognize signals from the microbiome, but also to communicate with the microbiome and to shape the composition of the microbiome. In a first step, Alexander did, together with Karolinska Institute, a single-cell expression profiling of these neurons and could identify 7 distinct neuropopulations in Hydra. Hydra has 3,000 neurons. We know every neuron by first name. And so we are at the moment in a different project setting together the whole nervous system. So there are 7 populations of neurons. And again, summary of this part, which was all published, this is the how question. How does Hydra contract spontaneously? And the answer is, there is this subpopulation N2, which are the pacemakers. They are characterized by these ion channels. They sit exactly there where our previous colleagues, wonderful old papers, 50 years old, have shown that if you cut out the tissue, then this contractile activity is gone, but not the movability of the whole animal. So this is the pacemaker population, and you can actually see that here. It's a beautiful neuron sitting around the head region. These are a few dozens of cells which do the whole job. I move now to the unpublished part, and actually not often you dare to address a why question. This is the most difficult question in evolutionary and developmental biology. Why is there a certain trait or a feature? But in this case now, it's very obvious that you may ask, why the hell do these animals have to contract all the time? Or why does this mouse gut here, does it continuously, and you put it in the aquarium and it goes on, it goes on, it goes on? For what reason? That was what Jana asked me in the coffee break at the Gordon conference 4 or 5 years ago. So we subdivided that question in 3 sub-questions. We first wanted to know if there is a direct link between the contractions and the composition of the microbiome. And that work was done by Christoph Gietz, graduate student in the lab. And what Christoph could show is, so what we did is we compared polyps which could contract and polyps which were under a controlled manner reduced in their contractions by giving some drugs. And by doing that, and then you did the microbiome signature sequencing, et cetera, and you can clearly see here from this PCA plot here, this is so that the, I have to move this, I have a strange interface here, so that you can see here that these mental treated animals, they cannot move at all. They have a very different microbiome from the other ones. The control is the gray one. Lidocaine leads to a kind of a minor disability to contract, has also already a very different microbiome from animals which cannot contract anymore. They are simply quite different. And it's not only that they are different, but also the abundance of certain guys in the microbiome. And we checked here Flavobacterium, Pseudomonas, and Acidovorax. changes compared to normal contracting animals. So bottom line, under normal contraction situation, 3 times per hour, you have a certain composition of the microbiome. But if you reduce that under a controlled experimental situation, you change the composition of the microbiome. And I can tell you that particularly microbes which are sitting normally at the foot region are now crawling up and colonizing the body column region. So you get a change in the microbiome when you disturb the contractions. The second sub-question on why do they need that or why do they do that was, now comes Jana into play. She was interested in what happens with the flow physics around the body column when the animals contract. And in this case, it's particularly significant for the microbiome because the microbes live just outside. So they face the water column directly, more or less. So here is the fluid flow physics story. And before I introduce you to Jana's data set, I have to introduce you to a very brief concept, a concept in fluid dynamics. And that is the concept of the boundary layer. In times when we could fly around the world, the boundary layer is pretty important for airplanes because the boundary layer is this layer of air which is in immediate neighborhood to the metal surface of the airplane. And the same is true for anything which is for a substrate or for an object which is in fluid. And so you have here the surface of the object, and then there is a layer of fluid which sticks to that surface. And then comes the free fluid. And the layer which sticks to the surface moves or does not move with the surface. It has no ability, there's so much viscosity there, it cannot move just on its own. That's the definition, in my words, as a zoologist and not a physicist, that's the definition of a boundary layer. I found a very nice picture which shows that on a human hand here. So the boundary layer is, and this is now in the air, it's the same thing. This is actually going around the body or here the hand. But of course, if you contract quickly, then you lose the boundary layer. That's the trick. So I tell you now what happens with these contractions, the animals lose the boundary layer. But the boundary layer is something very thick. And if you are never moving, this is not very healthy, particularly not for organisms which live close to the surface of the object. They cannot get whatever, chemicals, oxygen, nutrients. This is just a viscous layer around the surface. So I introduce you now to some movies, enjoy and relax. They go a little, and Jana Napwood has done them. We have submitted them together with the paper. And Jana, the movie is more or less self-explaining. And I show you 2 movies. And after these movies, I give you a summary of what we have learned from Jana's, from Jana's measurements. I go the wrong way. Here we are. So the movie has an explanatory text also, but what we test is the boundary layer formation in Hydra. And how a fluid physicist does it is she or he adds fluorescent dye to the animal. And what you can see is here that the dye goes to the animal, and then when it meets the surface, it just goes with the surface, and this tentacle will come down. And so then this dye just sticks to that. And so you can follow that and measure that. And I cannot speed up the movie. I'm very sorry. I tried that, but just enjoy. And then we look what's happened when the animal contracts. That will come in a few seconds. Also here, there's a little dye coming to the body column. So look, this dye is just sticking here and moving around here. So now the animal contracts. This is highly slow in motion now to let you follow what's happening. Here is the dye. And by contraction, the dye is shedded. We call it boundary layer shedding. And when the animal has contracted, which goes normally super fast, there is no dye at all. The dye is an indicator for the boundary layer. So that means after contraction, the animal has no boundary layer. And it can make a new one. This is fresh, which has nutrients and oxygens and chemicals and whatever. Okay. Now Jana measured the velocity of this fluid along the body column of Hydra with her fluid dynamic expertise and asked where is most of the velocity. And you see here, this is the Hydra head. When it contracts, it does an additional movement of a swirling around. And then the physicist can do all these measurements and can find so the velocity of this. And so she basically, she measured how quickly goes this boundary layer shedding at what distance from the surface. So this is the boundary layer, then comes the surface here. And under resting, of course, this goes very much slower, and there is actually no speed or no changing of the velocity there. And we found most of the movie goes on. Most of the movement is in the top, and the bottom stays stick. Remember, bacteria from the bottom crawl up at the body column when the animal cannot move. So the result of all these movement, of these measurements, Jana was several times visiting our lab and did the measurements, and then she did them in her own lab in Los Angeles. So in essence, that's the thing I want to tell you. So this is the fluid speed as a function of distance from Hydra's surface. And under a peak contraction, that means this short contraction, you have quite a change. Whereas this is the normal intercontraction interval, not much really happens. So the contraction events are really a perturbation, a quick change, but they are restricted to the upper part of the body, where most of the microbes live, and the foot remains more or less motionless. That means this contraction is actually a spontaneous and regular boundary layer shedding. The animals get rid of this sticky, viscous something, and they have the chance very regularly and continuously to rebuild that boundary layer. That could mean that this is, of course, a selected trait because it allows the animals to get nutrients, chemicals, whatever, also actually get rid of waste. To test if that is the case, we added another sub-question to our why question. And that brought Eva Kanzo into the team as a mathematician, and we cannot test it directly, of course, so we modeled it using a pretty challenging mathematical approach. We asked, do these contractions actually, are they important and beneficial for the animals in terms of could they enhance mass transport in field? That's what we thought they could, but we needed a model to say that at least in a model, this is reasonable. So I walk you through very slowly through this beautiful analysis, and this is a scheme of the animal, this is the head, and this is the foot. And normally you have this nutrient poor boundary when the animal is not moving. It has around the body, and particular on this elaborated head region, you have this what Eva calls a chemically depleted zone. And of course, that's about the whole animal. So now comes this spontaneous contraction, and our hypothesis is that this contraction is a reset for the boundary layer formation. And since the foot doesn't really do something, it's this chemically depleted zone stays in the foot, but in the head, it grows. You can model that, and this is shown here. So it takes a certain time in the model until this is the regrowth of this boundary layer. And in that state, it is probably of not much use to the animal anymore, because no chemicals and substances can actually get into the tissue. So we assumed that these contractions actually are a reset from here to here, and then it grows again, and that's the reason why these animals contract. So now Eva did simulation experiments. She simulated that approach thousands of times. In this experiment here, 10,000 times. And we gave different conditions, normal contractions, and then reduced contractions as with our experimental situation. So the data from the lidocaine, and then we also know that continuous light reduces the contractibility. And what she really could show is, so that the number of normal contractions is in her model 3 per hour. I come back to that. But most important, just look here, this growth of this nutrient depleted region again, that goes most efficient with the normal number of contractions, and 3 is the average number per hour. And of course, if you reduce the contractions, this growth doesn't really happen. That means the animals stick to their nutrient depleted layer, boundary layer, and have no chance really to get something new from the environment. That's the basic message of that model. And if you now go and put that into a kind of a new measurement, and then IFA comes to the conclusion that this 3 contractions per hour, which we also observe in the normal animals, is the most efficient way to increase this uptake. You can, if you do more of the contractions, you can increase this uptake even more. However, that comes at costs, because it's very energy costing. So this 3 hours seems to be a very good point. And if you reduce the contractions, of course, the efficiency of uptake is reduced. That was very interesting for us. And Eva invented this wonderful word. She said, this 3 hours, we always ask, I mean, many animals, the contraction frequency in different animals is different. And we ask, wow, Hydra is very slow, only 3 per hour. But with the mathematical model of Eva Kanzo, it seems that these 3 contractions per hour, what Eva calls the sweet spot of efficiency, this is just enough to get the enough and sufficient uptake by shedding the boundary layer. And if you do more, it would be much more energy intensive. And of course, reducing is not enough. So reducing is a problem. That's why these 3 contractions per hour may be selected for. So, in summary, I think we have found at least a partial answer to why do the animals do that. And the answer seems to be that they have to do it, otherwise they would be surrounded continuously by a sticky, viscous boundary layer, which of course is not visible to the normal eye. You have to be a physicist and you have to have all this technology to make it visible. It's a fluid, just water in our tanks. But in terms of physics, that's something which is very important to the animal, and that's why they do that. That's what we propose. because it then stabilizes the microbiome. The microbes have what they need, the epithelial cells have what they need, and if you disturb that, that's of no good. I come to an end and I want to summarize and I want to put it in a larger context. And again, thinking beyond Hydra. And very nicely, this movie Christoph gave me, there is a fantastic, fascinating paper from marine biologists in Woods Hole, and they discovered somewhere deep in the ocean a strange crab. And they call it dancing for food. And these crabs continuously, but slowly, I don't know how many thousand meters below, they do this movement. But if you read that paper, so these crabs harbor or farm bacteria outside because they need them for carbon fixation, because they live in this very nutrient-poor environment. And in the paper, they write, we hypothesize that this species waves its arm in reducing fluids to increase the productivity of its epibionts by removing boundary layers. Isn't that great? Published in 2011. These guys and these animals, you cannot do mechanistic studies. But with this simple freshwater polyp, I think you can manipulate it, you can reduce the fluctuation, and that's what we did. And so I come back to a statement which Margaret McFall and I made in a recent summary paper. The developmental biology of animals has been, the developmental biology of animals has been over evolutionary time shaped by interactions with the microbial world. And I think many of you and us, we believe that. The majority, unfortunately, of the developmental biologists has not yet got that message. We are working on that. I would extend that now from these new observations and say, it seems that these interactions with the microbial world, that is also not only true for the developmental biology of the animals, but also for the unconscious, spontaneous, and the conscious determinants of behavior. And with that, I be at the end of my talk. I think I have give credit to my collaborators. They did the story, not I. Jana Nafrot, Eva Kanzo, Christoph Dietz, Alexander Klimovic, and our collaborators for the single-cell transcriptomics. The work is supported by the German Science Foundation. This is the building and the center we are sitting in, and this is a neuro center I'm also a member of, and I acknowledge being fellow of the CFAR. And I thank you very much for your attention. I'm very open to questions.
Thank you so much, Thomas. Can I just ask you to maybe share so we can see more faces? And if you want to ask a question, please raise the hand electronically or write in the chat. I can see Ned and then Jonathan. So maybe Ned first.
Mas, that's fascinating work as always, and really a wonderful collaboration, shows the power of collaboration inside and outside biology. So my question is, would the prediction from the model be that if you increase the viscosity of the fluid outside, that there would be an increased frequency of the contractions? And is it possible that the animal is aware of what the conditions are at the surface and reacting to that?
It's an excellent question, Ned. We did not test that. Probably we could by changing the viscosity of the medium. And it's exactly as you say. I would expect that if you bring, actually we can do that going nearly to soft agar, but that would of course cause the animals to more heavily trying to contract.
It would be more work, yes.
Yeah, very good question. And do the animals know what they are doing? I don't know. But the nerve cells certainly realize what's going on outside. And so we still have not identified the chemical language between the neurons and the bacteria, but we are working on that. Yeah, I think it's the beginning of a story, and so thank you for the question. Yeah.
Yeah, thank you.
Jonathan from the IGC. You have to unmute.
Can you hear me now?
Yes.
Yeah.
Okay. So I found myself confused. I thought you were going to talk about the boundary layer inside the intestine.
No.
And so that made me ask, you didn't. But actually, I suspect that some of the same considerations would apply. Nevertheless, what you talked about in the latter part was the boundary layer outside. And just showing that I've never worked with Hydra and don't know anything much about it except what I learned at school, I didn't realize that they needed input, so to speak, from the outside. Chemical input. Are they're eating ions, eating small molecules and so on, or diffusing inwards from the outside surface. Is that correct?
Yes.
Oxygen.
Yeah, Jonathan. First of all, so this Hydra has a gastric cavity, but this is not a real gut. So the microbes are living in a glycocalyx, which is chemically very similar to our mucosa in the gut by mood genes and all these conserved components, and the bacteria living outside. And what we looked in, as correctly say, we looked into the boundary layer, which is outside the animal. And yes, this epithelium is very many molecules can be transported to that epithelium, actually even large molecules can be taken up. We tested that. There's early work. Small beads can be taken up from the outside. So there is a going in and out continuously. So yes, the environment plays a major role. And if you change the environment or if you make this inaccessible, this is a trouble for the animal.
But there are no surface adherent there's no surface adherent microbiota, external surface adherent.
Well, so these epithelial cells produce the mucosa, the glycocalyx, and the bacteria live in that Right. Okay, good. Thank you so much. I understand. Thank you.
Luís.
Hi. Hello. Unfortunately, I missed a lot of the talk because I made a mistake in the timing, but what I saw, I really enjoyed. So I thought it was very interesting, this modeling on the Hydra. I wanted to know, maybe you said it in the part I didn't see, so I apologize. It's just I wanted to know to what extent this behavior is observable across many species of Hydra that would live in completely different environment, maybe some of them where water doesn't move. So I think that would go a bit with the previous question, in places where the pressure is completely different. And when you try to use your model, do you find very different numbers of contractions per hour if you change these conditions? That is, because if you always find 3 hours, then you have to start wondering what's going on. And and the fact that the species happens to be at your sort of optimum, I was wondering to what extent, I'm always wondering when things work too nicely, if we shouldn't go back and try to understand what's going on. And finally, when I look at the curve that you are showing of the optimal, there seems to be a sharp increase at the beginning between 0 and 3 hours. But then after that, it was relatively flat going down. So the question is, how flat is that if you have a more realistic model? Because in a model, you always have to simplify. And maybe what's going on is that it's flat. So whatever you do, it could be any number, let's say above 2 hours. So twice per hour. So this is a bit about the modeling, the robustness, and the sorry.
Yeah, thank you so much, Luís, for the question. So I think I can answer the first two, and unfortunately, Eva is not here. I can try to answer the last one. of course, we did not try different species. I can tell you that from my experience with Hydra, so many different Hydra species are living, as you say, in different conditions, that we have so-called deep water forms, so they live much deeper, and others are more on the surface. In principle, they have conserved behavior, but nobody really checked the spontaneous contractions. Mm-hmm. And I would assume there is a green Hydra, for example, and anybody who has an aquarium at home, a freshwater aquarium, can see the animals are different. So not all species are the same, okay? We did not test yet what you and also NetRuby just suggested, experimentally changing the medium by higher viscosity, pressure. So we did not change that at all. Mm-hmm. That we come out with this number 3 was completely not planned and not expected. And actually, if at the beginning when she did the model, I didn't understand, but all of a sudden we realized, oh my God, that's the number what the animals really do. So it is, the model was not pressed into finding what nature has given us. It was the other way around. It's a coincidence that this model with these parameters gives this number 3 per hours. What we did is, as I showed, is that we change the contractibility of this so-called gut. That has, of course, in the model, that has then a real difference in terms of the predicted outcome. That's what we did. So the question, how does this curve then end, or how flat will that come? I simply cannot answer because I'm not a mathematician too deep in that model. I'm embarrassed that I present that now. Okay, thank you so much for the question.
No, thank you.
Miguel Soares from IGC. Unmute, Miguel.
I'm trying. Thomas, can you hear me?
Hi, Miguel.
Hi, Thomas. Very nice talk. I'm intrigued by a lot of things, but the one that really intrigues me is it's amazing that Hydra, to me at least, use neurons directly to sense microbes, right? So they seem to shortcut completely the need for more, let's call innate immune cells to do this job. My question is, do you know if these neurons express pattern recognition receptors that would help them sensing microbes? Yeah. And do you have any feeling of whether the neurons are interacting with, let's call it, macrophage-like cells that would help them do this job?
Yeah, thank you very much for the question. I cannot go back now to my talk, but actually, I guess the title of this PNAS paper, which shows the molecular architecture of these neurons, is actually neurons are immunocompetent cells. And the point of that paper is that our single-cell transcriptomics and all this analysis shows, to a big surprise to us, that 70% of this transcriptome is actually immunorelated. So they express all the TLR pathways and the MUMP receptors and so on, including effector molecules such as antimicrobial peptides. Many of them, some of them conserved, some of them what we call taxonomically restricted. And this is probably to be explained by the simple tissue architecture in evolution. So this is just an epithelium, ectoderm and endoderm, and the nervous system. They have invented the nervous system, and from the very beginning, they have no macrophages. The immune system is, people thought for a long time, is an epithelial-based immune system. What we find is that as soon as you invent a new organ, the nervous system, this system is integrated in what we call the meta-organism. It must interact also with the microbial environment. And neurons produce antimicrobial peptides, but we also have shown that neurons receive and are able to directly receive signals from the bacteria. I think this reflects the simple evolutionary position, and in more complex animals, this is getting more complicated. They have a much more elaborate immune system. Margaret McFall has a paper in 2007 in Nature, taking care of the community. And she proposed at that time, and probably at that time, nobody really took that serious, that the adaptive system, immune system of mammals has the same function as the innate immune system in invertebrates, meaning shaping, controlling, regulating the microbiome. And the mammalian microbiome is so much more complex than an invertebrate microbiome, so it needs an adaptive immune system. So the innate immune system in invertebrates goes beyond epithelial cells, and in animals which do not have immunocells, it is in all the cells present which are there. This is epithelial cells and neurons. There is no other cell type.
Thank you. Thank you very much.
We have Gonzalo, a student from IGC.
Hey, Thomas. Thank you for the amazing talk. So my question is I think, kind of simple. So we know gut pathogens, they interfere with contractions at the gut level, right? In different models. So I was wondering if you have any evidence, if that's the case in Hydra as well. And if you do, if that's, kind of like stopping the contractions would be kind of like a protective mechanism, because you're not renewing the environment, right? So it could be a defense against pathogens.
Yeah, thank you so much. Unfortunately, and that makes us, and you and Miguel and others always so, we have no pathogen. We have no real pathogen.
Okay.
I would love to have one. Of course, we can add some artificial stuff to the Hydra, but in nature, that was why I thought about the role of the immune system, because the animals invest so much in the immune system, and we have very little evidence for true pathogens. So this repertoire of immune molecules is just there to control the beneficial microbiome. Probably there are pathogens somewhere. We have none really available. I would love to have, but theoretically, yes, that could be, of course, a protection mechanism.
Okay.
Yeah, thanks.
Thank you very much.
Thanks.
Okay, I don't see any more raised hands. Oh, okay, I see now. Sorry, I don't know how to spell your name.
Yeah, I think that's probably me. It's Frauke.
Yes, it's you. Go ahead.
Yes, I was wondering, so do plants, aquatic plants, also have this problem that they get this boundary layer that's depleted of nutrients? And I'm not sure if you work with plants, but I would be interested in that. And how do plants cope with this problem?
That's a wonderful question. Of course, I love plants. I don't know much about them. I'm looking in my garden there, but plants are, in contrast to animals. If you ask a student what's the difference between a plant and an animal, one of the standard answers is animals are mobile and plants are sessile. And so animals have nerve cells and plants do not have nerve cells. I have no idea how plants solve the problem. Plants have an elaborate immune system, and colleagues such as Paul Schulze-Lever have shown in the last few years, also in our Gulbenkian summer school, that this immune system in the plants works very much as in animals as a shaper and a controller of the root or the leaf microbiome. But how that relates in water plants to what I just told you, the boundary layer shedding, I have no idea, and I don't think that anybody has, but maybe we should talk to plant people. Maybe they should look into that. Thank you so much.
Thanks.
Okay, Moisés from IGC.
Yeah, hi, nice talk. This might be a very stupid question. Maybe you already said something about that. But as far as I understood, these neurons are sensing the bacteria to trigger this contractility, right?
The bacteria produce something the other way around, Moisés.
Okay.
Bacteria produce something. It's a very small molecule. We don't have it yet. And this molecule is received by a certain subpopulation of neurons. Yeah. Then they get activated. We can do calcium imaging, and this activation activates other neurons, and then the animal contracts. That's how it works.
So, yeah, my question, maybe is, I mean, it's very stupid and not related, but to which extent what these neurons can also be sensing are the changes that are happening in the boundary layer?
That's a very good question. First of all, there is no stupid question at all. Every question is good, and that is a fantastic question, which we actually can address. Christoph is watching us, I guess, somewhere. I don't see him. And I come back to what Ned asked in the first question. I think because we can do calcium imaging, so we can directly check the activity of the neurons when the outside medium is different. Thanks for the great question. No answer yet, of course.
Okay, I don't see any raised hand at the moment, but I think we're getting close to our 3 p.m. Well, here in Portugal. It's very different times all over the world. We have Ned and Margaret that are in the middle of their night, right? But thank you so much, Thomas, for a great talk and beautiful questions and discussion. Thank you, everyone. It was a very exciting seminar. And for the participants of the meeting, please join us in the meeting link at 4 p.m. for the networking session. So you have a 1 hour break. It's a luxury. Maybe Ned and Margaret can sleep.
Thank you so much for listening. Thank you. Bye-bye.
Thank you, Thomas. Thank you so much.
Thank you. Bye.